In epigenome sequencing, we study epigenome. Epigenome is anything above genome level. That is, we are not interested in actual nucleotides, but we are looking into modifications on those nucleotides. And in addition to that, we can also study the modifications on the histones. So addition of methyl groups on cytosine residues, that is an important modification. So here we see that in the DNA, these cytosines having these methyl groups attached to them. And another thing to notice here is that these C's are then followed by G's. CPG islands, they concentrate more in the promoter regions, and in this way, with the help of them, we can identify the promoters, and we can also identify the presence of genes by identifying these CPG islands. And methylation in the promoter regions actually brings about the silencing of these regions. The analysis of these methylation patterns, we call it as methylome analysis. What we can do here is there is a technique called as bisulfite conversion. We can use that as on the whole genome, so we can call it as whole genome bisulfite sequencing. We can target to some specific regions, which we call it as targeted bisulfite sequencing. We can also capture this methylated DNA with a technique called as MEDIP-seq, that is methylated DNA immunoprecipitation. We can also do methyl cap-seq. So methylome analysis, as I said, that can be done with the help of bisulfite sequencing. We can also do restriction digestion based methods, and we can also do MEDIP-seq. In bisulfite sequencing, what we do here is we deaminate unmethylated cytosines. So once we put those, we remove the amino groups from them, and that is called as deamination. They are converted into uracils, and we can then convert those uracils into thiamines, and then we take those sequences, and when we compare them with those sequences where we have those methylated groups, we can identify those methylated cytosines. So in bisulfite sequencing, we take these regions where we suspect that methylated cytosines are present. So for instance, if we see here, we have this and this C which is methylated, whereas these two are not methylated. So we sequence them, and then we treat these similar molecules with bisulfite molecules. So what they do is they convert those C's which are not methylated into U's, and then these U's are then, when they are sequenced, they become T's. So the ones which are methylated, they remain as C's, but the ones which are not methylated, they become T's. So in this way, while comparing these two fragments with one another, we can identify where we do have methylation patterns which are right here in these two C's. In restriction based methods, we use some restriction enzymes. So those are the enzymes which cleave DNA at specific places. So these enzymes cleave the DNA where we have methylated nucleotides. So here we see one enzyme, MCRBC, so that cuts methylated DNA, and here is the segment which it recognizes. So it has purines, then we have cytosine which is methylated, and in between them there is some nucleotides which are 55 to 103 nucleotides are in between, and again we see this same pattern. So we have purines which may be A's or G's, and then again we have those methylated cytosines. So this enzyme actually recognizes these sites, and in this way we can identify those cytosines which are followed by purines and which are separated from one another by this much of length, and we can suspect that these cytosines are methylated. But this is limited. It provides only the data from those cytosines which are present where we have that restriction site. In MEDIP-seq, what we do here is we identify the methylated DNA by immunoprecipitation. So we take antibodies, and we design some specific proteins so which go there and attach to those cytosine residues that have been methylated, and then we design antibodies to capture them. So this is somehow related to another technique, ChIP-seq. So similar method is adopted. So in ChIP-seq, what happens here is we start with this DNA, so that has some proteins which are attached to it. So normally it's called as ChIP-seq because ChIP-seq stands for chromatin immunoprecipitation assay. So we identify those DNA fragments on the basis of the proteins which are attached to them. So this is interesting in identifying the DNA binding proteins and in identifying the binding sites for those proteins. So we shear that DNA, and then we add some specific antibodies to capture those proteins, right? So in this way we can capture the DNA and we remove those proteins, and then we submit that sequence for sequencing. So in this way we can identify what sequences are there, and we can have idea about those binding sites for these proteins. So far we have seen here is that sequencing of modified bases is mainly done in epigenome sequencing. Mostly we are interested in studying the methylation patterns in the DNA, and methylation is actually the key modification in the DNA.